So good afternoon, everybody. Welcome to our S2207 Kick Off. I'm Soni Smith, and I'm Vice Chair of the SWOG Lymphoma Committee, and I'm joined up here by our two co-chairs for this particular trial. Over the next hour and a half, we're going to go over some of the details and rationale, but just to sort of frame the discussion, diffuse large B-cell lymphoma we know is the most common lymphoma in the world. It's also the most common cause of death from lymphoma around the world. And although we cure a lot of people up front, this particular study addresses a very large population of patients where the frontline treatment does not work and aggressive therapies are not an option. One of the things I think we're really proud of from our committee is that this is a randomized trial. If you look at the 8 or so drugs that have been approved by the FDA or regulatory bodies around the world for lymphoma in the last several years, they've all been approved based on single-arm phase 2 data. And with that in mind, I think this is particularly a well-designed and important trial, and I hope all of you will support and participate. So welcome and thank you.
Thanks, Soni. Okay, so thank you for that introduction. So this is, as Dr. Smith mentioned, this is a randomized phase 2 study of the addition of targeted agents to tafasitamab lenalidomide in transplant-ineligible patients with relapsed refractory diffuse large B-cell lymphoma, or SWOG 2207. Let's see if I can get this to work. Maybe it doesn't work. All right, great. All right, so this clinical study will comprise really of two main components. It'll start with a safety run-in with the target population of relapsed refractory large cell lymphoma, and the primary objective of this part is to identify recommended phase 2 dose. And this will occur in the first 24 patients, 12 patients per arm, and this part will not be randomized, and we'll go through some more detail of this later. Once the recommended phase 2 dose is established, we'll move on to a randomized phase 3 study. Again, target population will be relapsed refractory large B-cell lymphoma, and the primary objective would be to compare the progression-free survival between the experimental arms and the control arms. So not between the two experimental arms, but with each experimental arm and the control arm. And this will be stratified by cell of origin and whether or not the patient was refractory to their last systemic therapy. Patients will be randomized 1:1:1 to arm 1, which is an experimental arm of tafasitamab lenalidomide plus tazemetostat, or tafa-len as a control, or tafa-len plus zanubrutinib. Okay, so as way of background, the majority of patients with relapsed refractory large B-cell lymphoma are not eligible for autologous stem cell transplant, representing a major unmet need. When considering CAR-T in second line, toxicities as well as access to care can be limiting, and many patients will relapse. So we will be focusing on older patients with relapsed refractory disease who traditionally have been exposed to several chemotherapy regimens, leading to large symptom burdens. There are frequent clinical and logistical barriers to more aggressive treatments often in these patients. And with this in mind, patient-reported outcomes will be essential to measure improvement in quality of life. The study will be based on tafasitamab or lenalidomide, or as we like to say, tafa-len, which is found to be an effective and well-tolerated chemo-free regimen. It was approved for the relapsed non-transplant eligible diffuse large B-cell lymphoma entity in July 2020. But we know that this combination doesn't really target specific biologic drivers of lymphoma. And large B-cell lymphoma, when classified by cell of origin through the WHO classification, is divided between germinal center subtypes and activated B-cell types. And there are certain biologic drivers that are thought to be responsible for some of these subtypes. So the study proposes adding biologic targeted agents in order to improve progression-free survival. And as mentioned, those two targeted agents will be tazemetostat and zanubrutinib. And I'd like to invite Hung-Lee Lee, one of our statisticians, up to go over some of the next points of the study.
Thank you, Dr. Amigo. I'm Hung-Lee Lee, statistician at the SWOG Study Center. Oops. Okay. Oops. As Dr. Amagor has mentioned, there are 2 experimental arms and 1 control arm. So the primary endpoint for randomized phase 2 study is progression-free survival. So it'll be compared between the experimental arms to the control. So specifically, it's tizamycin arm versus tafelen control, and a zanubrutinib arm versus control. Participants will be stratified by self of origin, as well as whether participants were refractory to the last systemic therapy. We assume 40% participants are GCB and 60% are non-GCB. The 12-month PFS in the tafelen control arm is 30% for GCB and 50% for non-GCB. With a sample size of 120 eligible participants in each of the control to the experimental arm comparison, there is 83% power to detect a hazard ratio of 1.75. So, sorry, this is corresponds to an improved 1-year PFS from 30% in the control to 50.3% in the experimental arm for GCB, and 50% to 67.3% for non-GCB subset. This also corresponds to an improved overall 1-year PFS from 42% to 60.5%. Secondary endpoints include PFS in GCB and non-GCB subsets, overall response rate, duration of response, and other survival endpoints in GCB and non-GCB, as well as in self-origin agnostic versus self-origin directed treatment cohorts. Exploratory endpoints include to explore PFS within subgroups defined by molecular profile and genetic subtypes, to assess frailty and symptom burden using cumulative illness rating scale and timed up and go tests. And we also assess its correlation with outcome. We're also interested in exploring immunomodulatory effects in tafelen arm and PFS in tafelen arm versus that in matched control, historical control from Almide and real-mind study. And now it turns back to Dr. Amagor for overview of treatment. Thank you.
Okay, thank you, Hung Lee. So first I'd like to go over some of the basis of why we chose some of these treatments. As we mentioned, tafelen is approved for relapsed refractory diffuse large B-cell lymphoma. Tafasitamab is a monoclonal antibody targeting CD19, and it was hypothesized that when combining with lenalidomide, it would increase natural killer cell activity, leading to increased ADCC and increased cell-mediated phagocytosis. So in the L-MIND study, which was a phase 2 non-randomized study, patients with relapsed refractory large cell lymphoma up to 3 prior regimens were included, and it did not allow for patients who were ineligible for transplant. Patients had to be ineligible for transplant, and patients were not allowed to be refractory, primary refractory to their last treatment. In the first 3 cycles, tafasitamab is given on days 1, 8, 15, and 22, and then on days 1 and 15 thereafter. And lenalidomide is given 25 milligrams PO per day on days 1 through 21 of a 28-day cycle. And this continued for 12 cycles. In this clinical study, if patients achieved stable disease or better, they were allowed to receive continued tafasitamab. I don't know how she got this to work. Here we go. So the combination was very well controlled, very well tolerated, with very few grade 3-4 toxicities. There was neutropenia, probably one of the most common toxicities. However, very few febrile neutropenic events, and thrombocytopenia is probably the other most common. Non-hematologic events included GI complaints, a little bit of rash being the most common. Impressively, in this chemo-free regimen, the overall response rate is 60% with a CR rate of 42%, and you can see the progression-free survival at 18 months is excellent. When thinking about the two experimental agents that we're going to be using, tazemetostat is an EZH2 inhibitor. This has been studied in a phase 2 clinical trial with 218 patients, where the main grade 3-4 toxicities included thrombocytopenia, anemia, and neutropenia. And you can see in the patients with diffuse large B-cell lymphoma, there was an enrichment of responses in those with EZH2 mutated subtypes with a response rate of 29%. However, in those with wild-type EZH2, there was a complete response rate of 8%. So there is an effect that occurs both in EZH2 mutated and wild-type, which may suggest that this could work in both GCB and non-GCB. I'd like to also point out that this tazemetostat is being studied currently with follicular lymphoma in combination with rituximab and lenalidomide, so very similar to the backbone that we're using here. And in the phase 1 portion of the study, there were no dose-limiting toxicities observed, there were no safety signals, and the recommended phase 3 dose was tazemetostat of 800 milligrams. The median duration of treatment for tazemetostat and lenalidomide was 9 months, although there were some frequent dose interruptions and discontinuation of one of these drugs. The other experimental drug will be zanubrutinib. This is a BTK inhibitor. And as you know, BTK inhibitors have been studied in the context of all kinds of lymphomas, including diffuse large B-cell lymphoma. Zanubrutinib is approved for other types of lymphomas, and in this phase 2 study dedicated to relapsed refractory diffuse large B-cell lymphoma, it led to an overall response rate of 29% and a CR rate of 17%, with a duration of response of 4.5 months and a PFS of 2.8 months. This was also well tolerated with few grade 3 adverse events. Most common were infectious and hematologic toxicities, and there was a very low incidence of rash. Interestingly, responses were enriched in CD79B and MYD88 mutated patients, which may suggest that this could have an enriched response in the non-GCP subtypes, and we're hoping to understand this a little bit more with our clinical study. I did mention that we'll be stratifying patients by cell of origin. This is patients won't be randomized to either experimental arm by cell of origin, but we'll be looking across the two cell of origins for a response to either of these experimental agents. And this will be done by immunohistochemistry staining using the Hans criteria by local pathology, and these pathology reports must be submitted within 15 days after registration. Some of the key eligibility includes histologically confirmed relapsed refractory large cell lymphoma, which includes both GCB and non-GCB subtypes, follicular lymphoma grade 3B, transformed lymphoma, and high-grade B-cell lymphoma with or without MYC, BCL2, and/or BCL6 rearrangements. As I mentioned, cell of origin determination must be performed by immunohistochemistry staining. Patients must have measurable disease with a PET scan performed within 28 days of registration. They must not be a candidate to receive autologous stem cell transplant, and this is something that's determined by the local investigator. But participants may have had a prior stem cell transplant or cellular therapy, such as CAR T. These patients are eligible. And patients may receive up to 5 prior therapies, and one must include a systemic multi-agent regimen for aggressive lymphoma. Some other key eligibilities: they must not have active CNS involvement, no known MDS or MPN or T lymphoblastic lymphoma or leukemia. Their prior therapy must have been completed prior to registration, and they must have recovered from any side effects. They must not have received prior tafasitamab, lenalidomide, BTK inhibitor, tazemetostat, and must not have any allergic reactions to these components. At the date of registration, they must not be taking any vitamin K inhibitors or moderate CYP3A inhibitors. And steroids are allowed for symptom control, but must be discontinued prior to cycle 1, day 1. Their performance status should be from 0 to 3. And finally, they must have adequate organ and marrow function. And you can refer to the protocol for sort of the definitions for this. At baseline, we'll be assessing history and physical exam, performance status, disease assessment, and toxicity. We'll be performing a timed up and go and cumulative illness rating scale. We'll have baseline laboratories, including CBC with differential, LDH, comprehensive metabolic profile, and pregnancy test. Patients will be imaged with PET-CT or CT chest, abdomen, pelvis, or MRI. Patient-reported outcomes will be formed prior to registration using the FACLIM and Pro-CTCAE, and we'll be submitting diagnostic tissue. And with that, I'll hand it over to Dr. Reagan.
Thank you. I'm going to do an overview of the treatment administration of the 3 different arms. First, we're going to talk about the control arm, which is actually arm 2, which is tafasitamab and lenalidomide. This is going to be given standardly. However, it will be a fixed duration, a total of 13 cycles. So tafasitamab for cycle 1 will be given on days 1, 4, 8, 15, and 22. For cycles 2 and 3, it'll be given weekly. For cycles 4 through 13, it'll be given every other week. The lenalidomide will be given throughout the duration of treatment, days 1 through 21 of each 28-day cycle. There are also dose adjustments in the protocol for patients who have kidney impairment. For experimental arm 1, tafalen and tazemetostat, it's the same dosing schedule for the tafasitamab and lenalidomide. However, these patients will receive tazemetostat for cycles 1 through 13, twice daily for days 1 through 28. The dose range on there is based on, we have to perform the safety run-in first, so the dose will be dependent on that safety run-in. For experimental arm 3, this will be tafasitamab, lenalidomide, and zanubrutinib. Zanubrutinib also will be given through cycles 1 through 13, twice daily, and that dose range again will depend on the safety run-in. For the treatment administration, as I alluded to before, this is a fixed duration therapy, so a total of 13 cycles of treatment. Tafasitamab, lenalidomide on the pivotal study, they gave the tafasitamab indefinitely if the patients remained in response or stable disease. Standard pre-medication will be used for tafasitamab, including acetaminophen, an H1 receptor antagonist, H2 receptor antagonist, and a glucocorticoid based on local standards. There's no pre-medication needed for tazemetostat or zanubrutinib. There's some supportive care recommendations in the protocol for nausea and vomiting, for diarrhea, and then for lymphoma-related symptoms, it's important to recognize that patients have to be tapered off of steroids prior to the start of cycle 1, day 1. We tried to be be overly prescriptive about steroids based on the population in the study. And then also some recommendations for tumor lysis prophylaxis in these patients. Some prohibited medications, so there is potential for interaction of tazemetostat and zanubrutinib with other administered drugs, including strong or moderate CYP3A4 inhibitors or inducers. No grapefruit juice, Seville oranges, or grapefruit. And then also to check frequently updated medical reference for a list of drugs to avoid. And there is a reference in the protocol. For our safety run-ins, our primary objective is to determine the recommended phase 2 dose. So again, the arms will be tazemetostat, tafasitamab, len, zanubrutinib, tafasitamab, len, and I think obviously the goal of this is to ensure the safety of the treatment combinations. We have some data of combining both of these agents with lenalidomide, but again need some more data of combining the 3 drugs together. There's 3 dose levels over on the right side of the slide for each of the treatment combinations. And there will be up to 12 evaluable patients per arm, so a total of 24 patients. Arms 1 and 3 will open at activation. Important to recognize there is no randomization for the safety run-in, and the participants can be enrolled onto the arms based on the investigator's choice. If there's slots, obviously. More information about the safety run-ins. So the dose-limiting toxicities will be assessed by the NCI CTCAE version 5.0. For cycle 1, patients will have weekly laboratory assessments and AE assessments, and then documenting the DLTs on the S2207 dose-limiting toxicities form, and documenting all AEs on the adverse event form. So a DLT definition, we'll go through that over in the table. So for hematological, any grade 5 toxicity, grade 4 neutropenia for greater than 7 days despite GCSF support, grade 3 neutropenia for greater than 14 days despite GCSF support, grade 3 or higher neutropenia with fever, grade 4 thrombocytopenia for greater than 7 days, grade 3 cytopenia with bleeding requiring intervention, and grade 4 anemia. And then other non-heme toxicities, so any treatment emergent adverse event leading to a delay in the initiation of cycle 2 for greater than or equal to 2 weeks, any grade 3 or higher non-heme toxicity, except for grade 3 AST or grade 3 ALT elevation of less than 7 days, nausea, vomiting, or diarrhea that persists less than 7 days. For cycles 2 through 13, laboratory assessments on day 1 of each cycle, AE reporting at the end of each cycle on the S2207 adverse event form. The MTD and recommended phase 2 dose will be determined based on the incidence of DLTs in cycle 1. All available safety and clinical activity will be reviewed to decide whether to explore intermediate dose and/or alternative dose regimens. Sites enrolling patients in the safety run-in must participate in mandatory conference calls twice a month on the second and fourth Monday. That's at 3 Central Time. We had our first one on Monday, as indicated in the protocol in sections 15.4. The site treating investigator and CRA or their designees will be required to attend the calls twice monthly from the time of the run-in participants' start of protocol treatment until the call occurring subsequent the participant's end of treatment visit. There we go. Some anticipated adverse events. For tafasitamab infusion reaction, for lenalidomide GI upset, cytopenias, and fatigue. For tazemetostat, anticipating anemia, nausea, and fatigue. And for zanubrutinib, anticipating some bruising, diarrhea, bleeding, fatigue, and infections. We skipped a slide. We're going to go back. We're having some malfunctions. So safety monitoring for all patients. So for every cycle except cycle 1, the following must be met within 48 hours prior to day 1 of each cycle. If the below parameters are not met, it will delay the start of the cycle until laboratory values have recovered. If delayed, then the laboratory values must be reassessed a maximum of 1 week later, but more frequent assessment is permitted. So for cycles 2 through 13, on day 1, platelets need to be 50, ANC of 1,000 or greater, a creatinine clearance of greater than or equal to 30 milliliters per minute, a total bilirubin of less than 1.5 unless patients have Gilbert's disease, hemolysis, or lymphomatous involvement of the liver. And if total bilirubin is elevated, then the direct bilirubin must be subsequently performed and resulted to be less than or equal to 1.5 of the upper limit of normal prior to the start of the next cycle of treatment. And then AST and ALT of less than or equal to 2.5 times the upper limit of normal. For serious adverse events, the study does require that expedited adverse events be reported to SWOG operations office using the CTEP AERS. Initially report on the adverse event form in the appropriate treatment cycle folder in metadata. The CTEP AERS report must then be initiated directly from the adverse event form in the metadata rave. And A's of special interest, T-cell lymphoma and abnormal bone growth. For dose modifications and interruptions, so dose interruptions, modifications, and discontinuations are allowed to manage toxicity. The doses will be modified as indicated in the protocol. This is in section 8.3. And then also in the following for lenalidomide in 18.11a, tazemetostat 18.11c, zanubrutinib 18.11f. There are no dose modifications recommended for tafasitamab. And for potential overlapping toxicities, there's a dose modification schema, tables 8.31 and 8.32. Maximum dose delay is 28 days. Missed doses will not be made up. If multiple toxicities are experienced, dose modifications will be based on the toxicity requiring the largest dose reduction. Whoop. And reduction, whoop. The reductions are based on the dose being given at the end of the preceding cycle and are based on toxicities observed since the prior toxicity evaluation. So once a dose reduction occurs, patients must remain on the reduced dose for the remainder of the study. Dose re-escalation is not permitted. If one drug is held or discontinued due to toxicity, 2 drugs may continue per protocol. If more than one drug must be permanently discontinued, the participant must be removed from the protocol therapy. And that's in section 7. For disease assessments, I think we had a lot of sort of internal discussions about this. And so our interim PET CT, our initial disease assessment, is around cycle 3, day 1. And I think we chose that because of the high risk in this population and the possibility that we may see relatively early progressions in patients. So we wanted to make sure we had a relatively early uniform disease assessment in these patients. There's additional disease assessment after cycle 6 is optional. We also do an end of treatment disease assessment, so all participants must have an end of treatment PET CT, and then 3 to 8 weeks after cycle 13, so if they complete the full year of therapy. And if the participant goes off treatment early, then 3 to 8 weeks after completion of the last dose. At the time of progression, patients must have a PET CT at the time of suspected disease progression, and then a biopsy to confirm progression is strongly encouraged. For follow-up, the follow-up schedule after discontinuation of protocol therapy and prior to progression, every 3 months in the first year, every 6 months for years 2 and 3 from registration, and CT scans should occur every 6 months. And follow-up after progression every 6 months for 1 year, then annually for year 2 and 3 after registration. The criteria for removal from treatment, so completion of protocol treatment, progression of disease or symptomatic deterioration, unacceptable toxicity. As I had mentioned before, if more than one drug must be permanently discontinued, treatment delay for all drugs for any reason of greater than 4 weeks, and participant obviously may withdraw for any reason. The participant may continue protocol treatment as long as the participant is continuing to clinically benefit in the opinion of the treating investigator.
Okay, so we'd like to invite Kat Gassick to come on up and talk about site initiation and registration.
Thank you. So in addition to the FACT limb and the Pro-CTCAE, there is the timed up and go assessment, which will have a protocol-specific requirement training. And that's available through CTSU under the protocol-related documents, and will be necessary in order to initiate the trial at your site. For treatment initiation, once the patient has been randomized, it can take no more than 14 calendar days after randomization in order for them to begin that treatment. This study, like many of our other protocols, is distributed by McKesson. So for all drug orders, please order through our drug order forms also on CTSU. And you just submit that to the McKesson website. It's all listed on the protocol contacts list. For the actual ordering, you should be able to submit that order and get next day shipping, but allow up to 5 business days total to actually receive that shipment. And we will probably release memos closer to Christmas and Thanksgiving in case anything changes. And go to the... Oh, and this is just more specific details on that, just what I said. The contact email there for ordering and submitting your drug orders is listed. Again, this is subject to change depending on holidays. And then for CTSU, when you actually do need to get that ordering form, it's a little tricky to navigate to it, but it's under protocol-related documents, not under the actual same place where you would find the protocol, but it's all available through CTSU. And then last note, lenalidomide is commercially available, so it will not be supplied through this mechanism.
Okay, and then we have Daria Shagina, who's going to be presenting the next few slides remotely, I believe.
Yes.
Okay, great.
You can hear me, right?
Yeah, we can hear you.
Okay. Um, my name is Daria, and I'm the primary data coordinator for S2207. This study is using Medidata Rave for data collection. At Data Management Center, we do not grant access to Rave. For access, site should contact CTSU Help Desk. Please also make sure that the delegation of task log is updated with a new task and re-signed by the principal investigator. The master form sets for S2207 can be found on the CTSU website. At SDMC, we strongly encourage oncology research professionals refer to CRA manual or RP manual, which is located at the CRA workbench for detailed guidelines for data submission. Data must be submitted for all participants, regardless their treatment status, including participants deemed to be ineligible after registration. Protocol section 14 provides a detailed schedule for data submission. Next slide, please. Patients registered in the safety run-in portion of the study will have the same schedule as patients in the randomized portions of the study. However, there will be additional forms for cycle 1, dose limiting toxicity availability form, and dose limiting toxicities form. They should be submitted within 15 days of completion of cycle 1 treatment. If participants deemed eligible and received at least 75% assigned dose of protocol treatment for cycle 1, they will be considered available for dose limiting toxicity. Next slide, please. Laboratory values form. For cycle 1 of the run-in, laboratory values must be assessed weekly prior to administration of protocol treatment and documented on day 1, 8, 15, 22, and in the end of cycle 1. Submission of data in a timely manner is critical for the safety run-in phase. Next slide, please. Specimen submission is required with patient consent only. There is an error in section 15 of current protocol stating that one stain slide is required if patient doesn't consent. That information is not correct and will be removed with the next revision. So only patients who consent should submit, should have specimen submission. Peripheral blood should be collected in 4 tubes, 10 milliliter each on day 1 of cycle 1 and cycle 2, and in the end of treatment. Diagnostic tissue should be collected before randomization and in the end of treatment, or at progression, if progression identified prior to end of treatment. Diagnostic tissue should be sent to the SWOG biobank within 28 days after biopsy. FFPE block is always preferred. All the specimens should be logged in the specimen tracking system. If specimen is not available, it should be documented in the specimen tracking system as well to get the expectation resolved. If later patient withdrew consent from biobanking, consent answers should be updated in the specimen tracking system too. If you have any questions for data submission, eligibility, please contact Data Management Center at any time, and we will be happy to help. Thank you.
And then we have Dr. Sater.
Hi, thank you, Jen and Patrick. My name is Haider Saeed. I'm a lymphoma doc at Moffitt. I'll be in charge of the patient-reported outcome objective. The reason we wanted to focus on patient-reported outcome is, as you've seen and as we alluded earlier in the protocol, that this is going to be focused on patients who are non-transplant eligible. Those patients in general, they're either elderly or have some comorbidities. So it's important to measure how does the intervention will affect their outcome, because there are several studies that shown in the upfront therapy that when the patient control their disease, they will actually report better outcome. However, there is lack of data in the relapse setting of such a data, and then so we wanted to focus on the patient-reported outcome in that setting. So the primary objective of the patient-reported outcome would be to compare the PROs through the FACT-LIM trial outcome index, which is a sub-classification from the FACT-LIM scoring system for PROs. We will be comparing that at 3 months after initiation of therapy, and will be between the control arm and both of the experimental arms. The secondary endpoint will focus on the total FACT-LIM score, which will include the FACT-G, which is a subscale from the FACT-LIM score. So the total number of questions the patient has to answer for this study is around 42 questions, which is the total FACT-LIM, and that one will have a subscale for FACT-G, will have subscale for the FACT-TOI. This is going to be done at the beginning of the study and at 3 months, and for secondary outcome and the primary outcome, we'll also look at the quality of life measurement from change from baseline to 12 months, especially for patients who continue to be on the study. And then we're going to measure another instrument, which is the PRO CTCAE. It's an NCI-developed regimen that basically tag along the regular CTCAE that's collected from the patient. But this one, we have a selection of 20 questions that are related to the classic side effects expected from the experimental as well as the control arm. So we'll be measuring those also to see if the patient will be reporting different set of side effects than we would be able to measure through the FACT-LIM. So the collection will happen at pre-registration, and then for the FACT-LIM, we will be collecting it every 3 months until the end of the study. And then the PRO CTCAE will be collected with each cycle, and the patient will hand it over as we see them in the clinic. Then we have the details in section 18.7 about the type of questions we'll be asking in the PRO CTCAE, and we'll have a training for those. Thank you.
I was going to review quality assurance and funding. So audits will be conducted at a frequency of 3 years, and a data safety monitoring committee will oversee the conduct of the study. Data safety monitoring committee members will receive confidential reports every 6 months from SWOG Statistics and Data Management Center, and will meet at the group's biennial meetings as necessary. The DSMC will be responsible for decisions regarding possible termination and/or early reporting of the study. And then in terms of funding, we do have federal funding for the collection of biospecimens at various time points, and these collections are mandatory. And then there's also some non-federal funding for additional administrative effort. This table just provides an overview of those funds. Everybody can examine that for a minute.
Okay, great. And now we'd like to invite Tricia Hernandez and Hilly Dillon to come on up to discuss some resources and materials for our patients.
Okay, I think
I think we have one before.
oh, did I go too far? This is very sensitive.
Yeah.
Is that the first one?
It's not moving. There we go.
Okay, there we go. Thank you. I just wanted to start by expressing my gratitude to the S2207 study team and Lymphoma Committee more broadly for their recognition of the challenges and burdens faced by patients with relapsed refractory disease and the distress after, in many cases, multiple rounds of treatment. We need more effective treatments with durable responses for these treatments. And as required by NCI and SWOG, Lymphoma Committee assigned research advocates, Hildy, who has actually been involved since the conceptualization of this trial and is moving into an emeritus role, and me as the new Lymphoma Committee patient advocate have been part of the protocol throughout its developments. As patient advocates, we have been able to provide input into protocol design and study design of patient-reported outcomes, as well as work collaboratively with Amanda Stamplis, the plain language specialist at SWOG, on the informed consent and patient language summary that I'm going to review in a moment. We are in full support of the protocol as relapsed refractory large B-cell lymphoma therapy options fail. Too many patients and better clinical and quality of life outcomes are needed. The impact of enduring multiple lines of treatment on these patients is felt in overall well-being, mental wellness, daily functioning, relationships with friends and family, work life, and can often result in significant financial hardship. We appreciate the study team's support of PRO analysis as patients with relapsed refractory disease are facing a challenging time in their cancer experience, and it is important to weigh the benefit of therapy both clinically and from a quality of life perspective. For some, this will be their first decision related to clinical trial enrollment. For others, they've been through trials before. Many are tired of multiple rounds of therapy and from the constant focus of cancer on their lives. Unless patients are proactively asked about their lived experience and with treatment outside of the clinic, they may not tell you. So asking open-ended questions can yield more comprehensive information about their experience. In addition to the patient language summary, I will now review, and that's actually available to you if you didn't get it on your way in. There are some copies that you can grab on the way out. I'm also going to share a financial resources sheet that's available for site teams. It's not a patient-facing document, but it's available for site teams to talk with patients about financial resources if that stressor is one for the patient. And so I will just go over the summary. Do you?
Yeah.
Okay, good. Let me see. Okay, there we go.
There we go.
All right. So really grateful to see these plain language summaries. It takes some of the complexities of the protocol that was just reviewed and really highlights those key issues that can be reviewed with patients who are making decisions to enroll in the trial. So you'll see in the summary, it specifies what the name of the trial is, and then it highlights those key areas, the purpose of the trial, why the trial is important, so the rationale behind the trial, who's eligible for the trial, and what treatments. And Amanda, is there another, or is this the only other slide?
There's another side, yeah.
So you'll note if you have the summary in front of you or if you get it on the way out, Amanda also includes graphics. So this really can help visually patients see the treatment options. Oh, perfect. Thank you. And it also talks about the randomization process and how long patients will be in the trial. It also talks about any related costs. And I'm excited to report that this summary will be available in Spanish as well soon, and that moving forward, all SWOG trials will have these plain language summaries available in English and Spanish to them. And then the other document we have is the financial barriers and resources that really just talks about some of the considerations related to what patients need to think about when they are considering enrolling in a trial, what costs they will be responsible for versus what is paid for by the trial, and then also a list of updated and current financial assistance programs that can be shared with those patients. And that's it.
Okay, well, that was really fantastic. And I just really want to thank everyone who's been involved with the development of this study. This really took a village, and everyone listed here had a very active role in participating in the design and in really getting all the nuts and bolts together behind the scenes. Our statisticians worked really hard with lots of feedback from the NCI and incorporating the PROs. Hung Lee, Mike LeBlanc, Ria Vida, and Joseph Unger really helped us a ton. Alexei Danilov helped with the translational design and really trying to incorporate some novel ideas that we might explore in the future. Haider Saeed really worked on the patient-reported outcomes and how to incorporate these in the study where it's going to be really important in patients where they're heavily pretreated. And one of our primary goals is really to ensure improved quality of life. We have an alliance champion, Dr. al-Baghdadi, who's been involved in a lot of the protocol design. Of course, our committee leadership, Dr. Sony Smith and Jonathan Freeberg, as well as Susan O'Brien, who really helped in kind of steering the direction of some of the ideas for this study. Our patient advocates, Hildy and Tricia, thank you so much for making sure that this is acceptable for patients and, patients will really benefit from this study. And then our SWOG staff. We could not have done this without you guys. There was a lot of negotiation with many drug companies and McKesson and patient samples that we really relied on everybody here to make this go forward. Chris writing the protocol, Jenny with all these negotiations, Kat for really getting this up and going, Daria. I mean, everyone was really involved. So I can't thank everyone enough. And also to our pharmaceutical partners. Again, we couldn't do this without you guys really having you believe in this study, and to provide drug for this study is really important, and McKesson for the distribution. Some of our contact information is here. You can really reach any of us anytime. We have a study chair email, S2207 chairs, that will reach all of us. You can also reach our data coordinator. The information is for the translational chair and patient-reported outcomes, and advocate in case there's any specific questions, but we're all available to help anytime. I think here's some additional contact information just in terms of specific questions that might come up during the conduct of the study. And with that, I think we'd like to open up for any questions. I do see there was a question already that was posted in the chat. Yeah, I think I looked at it.
I can read it.
Okay, great.
So first question from Christina Gardner. I'm always a little concerned when a protocol says to allow 5 business days to receive study drug. The patient needs to be started on lenalidomide commercially, yet treatment must begin within 14 days. Of course, we want patients to begin treatment as promptly as possible, but can you help us understand why the patient would not be appropriate to be on the study if it takes 15 days to start treatment?
Okay, so I can take this, I guess.
I think we can.
Yeah, so this has come up already so we have one patient enrolled, which is super exciting. And, sometimes lenalidomide, sometimes we can get it immediately, and sometimes it takes a little time, depending on patient's insurance. And so we have been kind of kicking this around for a while, and our initial thought was 14 days would allow enough time. I know the site that's participating, they got it right in the 14 days. And one thing that we were considering including into the protocol, and we'll see how this plays out with the next few patients enrolled, is if we want to have almost like a longer window specifically for lenalidomide, so patients get started, and if it takes another day or two, then they start the lenalidomide a few days later. But this is something that's open for discussion right now. One other consideration is if you identify a patient that you're thinking about, to start the process early in terms of enrolling in the REMS program and getting insurance approval. So they don't need to be registered for the trial to get that component going quickly.
Just a couple things that I would add about that. So I think, as I've been thinking about how we put patients on this trial, I think that, if, let's say, they end up screen failing, I think that, a rational plan would be to treat them with lenalidomide and tafasitamab off study. And so I think as that patient signs consent, I think starting that process obtaining lenalidomide as early as possible would make sense. And then in terms of the timing, I think that a concern in this population is that if we wait too long to initiate patients on study treatment, we're going to have patients who drop off because of rapid disease progression. So we did want to have wanted to get patients started relatively quickly after they've been registered. Looks like we have another comment from Daria. While completing eligibility checklist is open, projected start date should be indicated within 14 days. If for any reason cycle 1, day 1 is delayed, that would be a minor protocol deviation, but patient will continue on study.
Any other questions or comments? Yeah.
Yeah, thank you for the presentation, very helpful. I see that the study was activated on June 30th. So what's the status of the study right now?
Well, we have one patient enrolled who started treatment, I think, about 2 days ago. Yeah. And so it's active. So it's in the safety run-in portion right now, which is not randomized, which means that you can enroll patients based on your choice. And perhaps, you know, if you have a preference between using zanubrutinib or tazemetostat, patients can be enrolled to tafelen plus either one of those experimental agents.
Thank you.
Lots of spots left.
All right. Well, if you have any questions, we're here. And thank you, everyone, for participating in the presentation.
Thank you.